So good morning to all of you, and I welcome to my lecture on BTL 410, Introductory Biotechnology and Genetic Engineering. In my first lecture, I discussed with you the basic concepts of biotechnology and genetic engineering, and also the scopes of biotechnology and genetic engineering in agriculture and the overall life sciences. Today we shall discuss on DNA and RNA, how protein is synthesized in the cell through the expression of genes and some other fundamental aspects, the structure and functions of DNA. Let me show you some of the slides, and it would be an overview for your knowledge that you received in biochemistry. I guess all of you received courses from biochemistry, right?
Yes, sir.
Okay. So today we shall discuss and mainly focus the structure and functions of the blueprint of life. That means DNA and RNA. They are considered the blueprint of life because mysteries of all activities in the lives of all organisms are written in the code form in the DNA and RNA. Is a slide changing?
Yes, sir.
Okay.
Sir.
All living things are made of water, an inorganic compound. Other inorganic compounds, mostly salts. This is why it is thought that life originates from the water, that is from the ocean. Organic compounds contains carbon and hydrogen. Any compounds we call organic if they contain carbon, hydrogen, and oxygen, because organic compounds, they are derived mostly from the carbohydrate biosynthesized through the process of photosynthesis. So all living things are made of water. Other inorganic compounds, water is an inorganic compound, lots of organic compounds, carbohydrates, lipids, proteins, and proteins are most interesting, and nucleic acids. Nucleic acids is the, as I mentioned, the blueprint of life. When we look at a living thing, what we can see? We mostly see the protein. So how does an organism produce its particular protein is a fundamental question. As in people protein versus tree protein, there are some differences. So the answer is DNA. How protein is synthesis? Protein is synthesized by the instruction of DNA. What is DNA? DNA is a nucleic acid, deoxyribonucleic acid. The species particular DNA sequences produce the species particular proteins. That means DNA sequences are unique for every individuals. Genes code for protein. What are genes? Genes are small fragment of DNA sequence which code for protein. Genes are long strands of DNA on chromosomes. That means in the DNA, there are small fragments. You can see here in the chromosome, DNA locates in the chromosome and gene locate in the DNA. So if we academically say, what is nucleic acid? Nucleic acids are macrobiomolecules, occur in every living cell, where they not only are responsible for the storage and transmission of genetic information, but also translate this information for a synthesis of protein characteristics of the individual cell. The nucleic acid makes up the blueprint of life. Nucleic acids are highly organized polymers of nucleotides, which are united by phosphodiester bonds. So nucleic acids are polymeric substances, and the unit of nucleic acid is called nucleotide. And each nucleotide is connected to another one by phosphodiester bonds. We shall get more detail step by step. What is DNA? DNA is the genetic code, instructions for heredity, composed of genes, director of protein synthesis, and DNA is also a type of nucleic acids, a type of nucleic acid, a type of organic compound, because DNA is biosynthesized in the living cell. A polymer, a compound made of repeating subunits, is called polymer, a polymer of nucleotides. What DNA stands for? DNA, already I mentioned, deoxyribonucleic acid, consists of ribose sugar with a missing oxygen. That's why it is called deoxy. And it's found in the nucleus of eukaryotic organism. Here is the illustration. You can see where does DNA locate. Where does DNA locate? If I ask you this question, the answer would be DNA locates in the nucleus. And more specifically, DNA locate in the chromosome. And chromosomes are located in the nucleus. Here you can see DNA double helical structure, and chromosomes you can see in the nucleus. Now the question is, does DNA locate only in the nucleus? In case of eukaryotic organism, DNA locates not only in the nucleus. For example, human, plant, and other eukaryotic organisms, DNA locates also in the mitochondria, and small mitochondrial DNA are also there. So mitochondria also have DNA. Nucleus also has the DNA. In case of plant, DNA also locates in the plastid. So if I ask you where DNA locates in plants, the answer should be mostly in the nucleus, mitochondria, and also in the plastid. How does DNA code for protein synthesis? This is very fundamental question and very important, and you need to clearly understand. DNA code for the protein synthesis. How? Very interesting. Certain gene in the gene segment of DNA express means gene expression actually associated with the synthesis of the protein in the cell. So how it does? DNA, transform into, and due to gene expression from the particular gene, specific mRNA is synthesized in the nucleus. It is called transcription. DNA transcribed into mRNA. mRNA means messenger RNA. And then messenger means it carries some message from the gene, and then it moves to ribosome in the cell. That means come out from the nucleus, and ribosome present in the cytosol, cytoplasm of the cell. And ribosome, you must remember it. Ribosome is the site for protein synthesis. When messenger RNA carries the message and comes to the ribosome, then depending on the information it carries, it recruits different amino acids. There are 20 essential amino acids that are involved in synthesis of protein, and protein is also a polymer of amino acids. So the recruitment of amino acids from one by the help of tRNA at the ribosome results synthesis of protein, and it is called translation. So transcription means when DNA transcribed into messenger RNA, this process is called, step is called transcription. And then it moves to ribosome and synthesize the protein as per instruction carries by the messenger RNA. And this process is called translation. So protein is synthesized by the instruction of DNA, and the site of protein synthesis is ribosome. And protein synthesis occurs mainly 3 steps. First is DNA transcribed into mRNA, and then it is called transcription, and then mRNA moves to ribosome, and when it moves at the ribosome, it recruits, depending on the information it carries, the amino acid and make a polymer of the protein. And each protein is unique, and it is called translation. So DNA and protein synthesis then happens this way. DNA sequence codes, how letters are put together, produce messenger RNA sequence codes. So DNA sequence code is transformed into messenger RNA sequence code. And messenger, RNA is single-stranded, but DNA is double-stranded. And then the messenger RNA leaves the nucleus and attaches to a ribosome in the cytoplasm, I already mentioned. Third, transfer RNA brings amino acids to the ribosome to match the sequence codes. Here is the ribosome, the structure, my left-hand side.
Second point আর স্লাইডটা একটু আরেকবার বললে ভালো হয়, এর আগে যেটা পয়েন্টটা বলেছিলেন।
Second, the messenger RNA, mRNA, leaves the nucleus and attaches to a ribosome in the cytoplasm. A ribosome means there are lots of ribosomes, okay? And then at the ribosome, there are transfer RNA. So transfer RNA brings amino acids available in the cytoplasm one by one, depending on the code of the messenger RNA, the message it carries, which amino acid should be recruited after one by one. Here is the ribosome where you can see here transfer RNA recruit the amino acid. So protein is synthesized, and protein synthesis means the expression of DNA. If I ask you what is gene expression, gene expression means gene transcribed into mRNA, and mRNA moves to ribosome and synthesize a protein, and protein actually does the job. All responses we see You feel cold, you feel warm, even you feel pain. Everything is associated with the expression of the gene and synthesis of the protein. So summary is DNA is a nucleic acid. DNA stands for deoxyribonucleic acid. DNA, a polymer of covalently linked deoxyribonucleotide units, lack of oxygen at 2' of its ribose sugar. So what are the major functions? DNA is the chemical basis of heredity because you are Homo sapiens, for example, human baby is like human baby due to the transfer of the genetic character from parents to the offspring. So it is the chemical basis of heredity because when a cell is divide, then DNA also needs to be copied, and then new cell also get the same DNA and may be regarded as the molecular repositories of genetic information. DNA is exclusively responsible for maintaining the identity of different organisms over millions of years. For example, Homo sapiens, or cow, or tree. Rice, for example, rice, Oryza sativa, maintaining its identity as Oryza sativa due to the DNA inherited from the parents to the offspring. There are another cousin of DNA. A cousin of DNA is called RNA, ribonucleic acids. Nucleic acid, ribonucleic acid, RNA. mRNA, there are several types of RNA. For example, mRNA, rRNA, tRNA. mRNA carries genetic information from DNA to the site of protein synthesis. So the site of protein synthesis in the cell is ribosome to specify the sequence of amino acids. rRNA plays a role in the binding of mRNA to ribosomes and in the protein synthesis. rRNA forms a workbench along with mRNA. rRNA and mRNA make a workbench to for the synthesis of protein at the ribosome. tRNA delivers the amino acids to ribosome for protein synthesis. That means tRNA, transfer RNA, pick up right amino acids as per the instruction carried by the mRNA, and one by one supply to the new synthesis of a protein as instructed by the DNA. So RNA as ribonucleoprotein, participates in the post-transcriptional modification of other RNAs. So RNA is the genetic material carrying hereditary information in many viruses. Can you name a virus which contains RNA? Can anyone?
Yes, sir. Coronavirus.
Coronavirus. Do you know what is the name of the coronavirus we are suffering, from a pandemic? COVID-19 causing coronavirus.
COVID-19. COVID-19. SARS COVID-19.
No. COVID-19 is the name of a disease.
Omicron. Omicron.
No. What is the name of the virus which is causing COVID-19 disease?
SARS-CoV-2.
Excellent. SARS-CoV-2. What SARS-CoV-2 stands for? Severe acute respiratory syndrome coronavirus 2. There are lots of coronaviruses, and SARS-CoV-2 is belonging to the group of beta coronavirus. So SARS-CoV-2 is an RNA virus. It contains RNA which encoded covered by, different proteins, and a coat protein like spike protein, and the structure, the peculiar structure, a crown-like structure. Okay. So nucleotide is the unit of RNA and DNA. Both are polymer, called nucleotide. Nucleotide is the phosphoric acid ester of nucleoside. Nucleotide have three characteristic components. One is a nitrogenous base, a pentose, and one or more phosphates. Nitrogenous base. What are the nitrogenous bases involved in synthesis of protein, synthesis of DNA and RNA? The nitrogenous bases found in nucleotides are aromatic heterocyclic compounds. You can see here purine, pyrimidine. So the bases are derivatives of two parent compounds, namely purine and pyrimidine. Purines are numbered in anti-clockwise direction, while pyrimidines are numbered in the clockwise direction. This is the internationally accepted system to represent the structure of bases. Major bases in the nucleic acids are DNA, RNA both contain two major purine bases. One is adenine, another is guanine. DNA, RNA also contain two major pyrimidine bases. In both types of nucleic acids, one of these is cytosine. But difference in DNA and RNA is that DNA contains thymine, whereas RNA contains uracil. What does it mean? DNA contains adenine, guanine, cytosine, and thymine. And RNA contains adenine, guanine, cytosine, and uracil. So difference is uracil and thymine in the structure, especially the bases. Sugar, also two types of sugar involved in the structure of DNA, RNA. DNA contains deoxyribose. RNA contains ribose, beta D ribose. Here you can see the differences in the sugar, a 2' OH here, and here 2' H. In case of DNA, deoxyribose. That means one oxygen is lack in case of DNA compared to the RNA. So we can review the location of DNA in the cell very clearly, where DNA locates. If it is a cell, there are some, thread-like structure in the nucleus called chromosomes. And in the chromosome, there is double helical structure DNA. The complete set of human chromosome, do you know how many chromosomes, pairs of chromosomes are in our DNA nucleus? We have 23 pairs of chromosomes, and human chromosomes, you see, unusually varied structures, and only one pair, X and Y chromosome, and all others you can see different structures. And XY chromosome is associated with the gender of human. So chromosomes and DNA replication is very important. The nucleus of one human cell contains approximately 1 meter of DNA. Very interesting. So if DNA molecules one by one is connected, then it would be 1 meter length. But you cannot see a cell as a human cell in the naked eye. There is another interesting point: histones. DNA, how DNA remains in the nucleus. DNA tightly wrapped around a protein is called histones, and nucleosome, and it is called nucleosome. That is when DNA, here you can see inside the 3D structure of histone proteins, and DNA is encircled and tightly wrapped on the surface of the histone protein, and histone plus DNA is called nucleosome. So DNA replication, DNA can make its own copy. So how? First DNA unzip, and then complementary copy of the single-stranded DNA is synthesized, and new DNA is produced. And DNA replication must occur before a cell divides, because new cell must have, the daughter cell must have the same DNA. And each new cell needs a copy of the information in order to grow. Here is the very simple diagram that showing how DNA is copying. And parent DNA double helical structure, here you can see. And template strand is, when it is unzipped and single strand is formed, and then new strand is synthesized, which is complementary to the single-stranded one, and make the two DNA. You must know that in case of the making the sequence, ATGC, adenine bind with thymine, and guanine bind with the cytosine, and there are some reasons. RNA very similar to DNA, and exception is, in case of RNA, ribose is the 5-carbon sugar. Uracil replaces thymine, and RNA is single-stranded. If you are asked what are the major structural differences between RNA and DNA, RNA is contains the ribose sugar, DNA deoxyribose, and uracil replaces thymine. That means in DNA thymine, and in RNA uracil, and RNA is single-stranded. And here you can see the double-stranded structure of DNA. The size of DNA, can you guess? The diameter of DNA, the length is, I told you, all DNA, if connected one by one in human, it would be 1 meter long. But you cannot see in the naked eye, because DNA is, size is nanometer. For example, its diameter is 2 nanometer only. 2 nanometer means very small, and you cannot see even by using light microscope or compound microscope. To see the DNA double helical structure, you need to use transmission electron microscope, TEM. DNA deoxyribonucleic acid, carrier of genetic information, figure showing 2 polynucleotide chain are held together by hydrogen bonding. Here 2 polynucleotide chain are held together by using hydrogen bonding between the bases on the different strands. So hydrogen bonding hold the 2 stranded together. And do you know who determined the double helical structure of DNA? It is a historic discovery which significantly promoted the understanding of the life science. James Watson and Francis Crick in 1953, they for the first time proposed the double helical structure of the DNA, and they analyzed the DNA by various ways, but they were not confirmed that the DNA is a double helical structure. 2 other scientists, Rosalind Franklin, very famous in molecular biology, the lady, and Maurice Wilkins, they used X-ray, they passed X-ray through the DNA, and their hypothesis was, if DNA is double helical, then the X-ray image should be X-shaped, and it happened. The right side you can see the image, and that helped Watson and Crick to confirm and propose the double helical model of DNA. But Maurice Wilkins James Watson, Francis Crick, they got the Nobel Prize. Actually, James Watson was a young PhD student working under Francis Crick at that time. And Rosalind Franklin was his friend. And Maurice Wilkins and Francis Crick, they are the senior professors in UK. So three guys, Watson, Crick, Wilkins, they got the Nobel Prize for this discovery, but Rosalind Franklin did not get it because just before the declaration of Nobel Prize, she was died, unfortunately, due to breast cancer. So in 1962, Nobel Prize in Physiology and Medicine was owned by James Watson, Francis Crick, and Maurice Wilkins. What about Rosalind Franklin? Unfortunately, she didn't get because she died. And in case of Nobel Prize, there is no posthumous system for awarding. And this is the simple paper, Molecular Structure of Nucleic Acids. It is 1.5 pages, which actually revolutionized the molecular biology. The Watson and Crick model of DNA structure. Here it is the description. Possibly you studied in your biochemistry class. I am not going through, line by line, and you can get all the information. So the chemically structure is like that. Here you can see the thymine and adenine, they are connected by hydrogen bond. And these two nucleotides, they are antiparallel, and hydrogen bonds are holding them in double helical structure. So DNA and its binding blocks, already I told you, binding blocks are sugar, phosphate, base, and nucleotide, like this way. Here you can see phosphate group with sugar and a base is called nucleotide. And this nucleotide is the unit of DNA and RNA. And they can make copy in the cell. And more interestingly, we can make copy of the DNA or DNA fragment in the laboratory. Do you know what is the process or a technique we use to make the copy of DNA in the laboratory? What is the machine or process? Do anyone have any idea? Any student? No idea?
Sir, use PCR.
Yes. By using PCR, polymerase chain reaction, Mullis received Nobel Prize for that discovery. We can make the copy of the DNA. For example, you have a small fragment, you can make millions of copy of that fragment, like photocopy machine. So PCR, using PCR, we can make many copies of the DNA. And the scientists got the idea from the natural system. So building blocks of DNA, you can see the yellow circle like a phosphate, sugar, and a base, for example, adenine or like that, and make the nucleotide. And this is the unit. So one of the interesting point is guanine and cytosine content. Guanine and cytosine content, when we do the whole genome sequence of an organism, then we estimate the guanine and cytosine content in the DNA because it is unique for different species. Generally, GC is about 50%, but extremely variable. For example, slime mold GC content. GC means, adenine, guanine, cytosine, thymine. Among 4 nucleotides bases, GC content, slime mold 22%, mycobacterium 73%. So you can imagine how different. Distribution of GC is not uniform in the genome. So different genomes have different GC content. So it is one of the unique character for the species. Interestingly, why adenine bind with pairing with the thymine, and why guanine pairing with the cytosine? It is a question. Adenine and guanine are bigger because they both have two rings. Here you can see adenine and guanine. Adenine and guanine both have two rings in the structure, but cytosine and thymine single rings. Cytosine and thymine only have one ring. So here you can see smaller base with the larger base, how they form the hydrogen bonding. So hydrogen bonding with a smaller base is with the paired with the bigger base. So the question in your exam you may find, why adenine pairs with thymine, guanine pairs with the cytosine? These particular pairs fit exactly to form very effective hydrogen bonding, as I have shown here, very effective hydrogen bonding in case of these pairs. These hydrogen bonds holds the two chains together in a double helical structure to keep the two chains at a fixed distance from each other all the way along. What forces affecting the stability of DNA double helical structure? It was a big question how this double helical structure is stable. There are some forces. For example, hydrophobic interactions. There are hydrophobic interactions which stabilize hydrophobic inside and hydrophilic outside. In case of DNA, DNA inside is hydrophobic. That phobic means dislike water, and hydrophilic means like water. So there is a balance and stabilize. Stacking interactions, it is also stabilize the structure. Relatively weak but additive van der Waals forces are work here. Hydrogen bonding also stabilize, relatively weak but additive, and facilitates stacking of these two in the ladder, two sides of the ladder. Electrostatic interactions destabilize, contributed primarily by the negative phosphates, affect intra-strand and inter-strand interactions. Repulsion can be neutralized with positive charges. Positively charged sodium ions or proteins, they are participates for the stabilization. So electrostatic interactions, hydrogen bonding, stacking interactions, and hydrophobic interactions, some stabilize, some destabilize, and finally hold the double helical structure in a stable form. Overview of DNA structure, already I told you, not repeating. These are no meaning to repeat. All cells replicate their hereditary information by templated polymerization, as I told you. Already I mentioned transcription. DNA is copied into mRNA with the aid of RNA polymerase. If I ask you which enzyme is involved in transcription of DNA into mRNA, RNA polymerase. The RNA polymerase will bind to the promoters that act as signal in the DNA sequence to make RNA. There is a promoter, a region in the DNA. So this is the transcription continues. Here you can see how transcription is continuous. First, double-stranded DNA needs to be single-stranded, and then you see guanine, cytosine, adenine, thymine, they recruit and make the double-stranded DNA. So exon and introns, there are some other terms in molecular biology. Exons means a segment of DNA in eukaryotic organisms that codes for a specific amino acids. That means exons is very important, which give the code for recruiting the specific amino acids for during protein synthesis. Introns, a segment of DNA that does not code for amino acids. That means in the DNA there are different segments. Exons codes for amino acids, Introns not code for amino acids. So non-coding region also present in the DNA segment. So DNA segment, all the sequences are not involved in coding. Exon, for example, exon 1 and exon 2, there is an intron. So intron during protein synthesis, mRNA synthesis, they can be spliced. All cells transcribed portion of their hereditary information into the same intermediary form, mRNA. And this is the summary of central dogma from DNA to protein, how gene expression taken place. We call gene expression. That means gene expression means specific portion of the DNA, that means gene, when transcribed to produce the RNA, called mRNA is called transcription, and then RNA moves to ribosome and synthesize the protein is called translation. So, and this is generally called central dogma, and you must remember this central dogma. And here you can see gene expression. For example, gene A, gene B, gene C. There are lots of genes in the, for example, human genome. 40,000 of genes are being, identified. So gene expression means synthesis of the, ultimately synthesis of the protein, not directly first mRNA and then synthesis of the protein. Protein 1, protein B, protein A, protein B, protein C, they are involved in the function of the cell. So transcription, a process by which a DNA template is copied to an RNA strand. Transcription is the enzymatic process where RNA polymerase is involved, and genetic information contained in one strand of DNA is used to specify a complementary sequence of bases in an mRNA, and then mRNA produce the protein in the ribosome. The fragment of genetic information corresponding to one protein is known as one gene. So I am going to end of the talk. Genetic code, another term. In the flow of genetic information in living organisms, the language of all the molecules should be read by enzymes or any concerning reader. DNA could be read by bases. Proteins could be read by amino acids. mRNA could be read by what? It will be read by codon, because in mRNA there are some codon. Codon means 3 letters in the mRNA. They are called codon, and scientists discovered the genetic code and got the Nobel Prize. So evolution of genetic code, Greek first genetic code evolved from the simpler form of the encoded fewer amino acids. There are lots of people worked to confirm the genetic code. So codons means a 3-letter word that specifies an amino acid present in the mRNA. So 3-letter words, for example, AUG or ACA. So adenine, cytosine, adenine, and it recruits a particular amino acids. Adenine, uracil, guanine recruits particular amino acids, and it is called codon. So mRNA carries lots of codes, and each code is called codon. And depending on the code, the protein is synthesized. So genetic code, poly AUC and G, mRNA provided information of amino acid coding. So every code, 3 letters code, is responsible for recruitment of each amino acid. Scientists discovered all the mysteries, and there are already, as you know, in the mRNA, there are only 4 letters. What are that? Adenine, guanine, cytosine, and uracil. So from 4 letters, different combination of 3 letters made lots of codes. But there are some code, for example, UAA, UAG, or UGA, they are called stop codon. They cannot involve in synthesis of the protein. So the set of triplet code words in DNA or mRNA coding for the amino acids for protein is known as genetic code. So today I should stop here up to the genetic code, and if you have any question, you can ask, and this is your time. So please ask a few questions. If you do not ask me question, then I shall ask you a question.
Sir, is it necessary to practice all of the figures that you have showed in the slides?
You have already studied in biochemistry. For understanding the biology, mystery in the biology, biotechnology, I repeated, lots of the, information so that you can have a clear picture. And in biotechnology, you must understand the structure of structural differences and uniqueness of RNA, DNA, and obviously how proteins are synthesized in the cells, as well as genetic code, codon, and gene expression. Gene expression is directly, gene expression means transcription and translation and synthesis of the protein by the instruction of the DNA. So I showed lots of slides due to give you easier story, but I could tell you within 10 slides all the stories. So many of the things are repeated. Go through and try to understand, not to memorize. For example, if I ask you chemical basis of DNA and RNA, so you can easily tell in case of DNA, there are adenine, guanine, cytosine, thymine, but in case of RNA, instead of thymine, there is uracil. DNA is double-stranded, RNA is single-stranded, or like that. DNA contains a deoxy ribose, or RNA contain ribose sugar, like that. So I showed lots of slides for your understanding, and many of the slides are, just review one as almost the same. So make your own story. In the books, they are very tightly, so it would be very difficult for you to understand. Any more question? Honestly, thank you. Do anyone have any question? Kanis Fatima June?
Sir,
Yes, who was speaking?
আমি স্যার এই জিসি ডিস্ট্রিবিউশন ইউনিফর্ম না হওয়ার মানে কারণটা কি এখানে?
Very interesting. If you ask me why different species have different DNA sequence, I cannot answer. Through the evolutionary process from the start of the life, each species has some unique properties, and they evolved in different ways. So in the phylogenetic tree, you can see incredible diversity in the living organisms. So GC content, why one species to another different, really tough to answer, because through the evolutionary process, when organisms evolved, and organisms are always evolving through the process of time, for their own fitness to the ecosystem they are living, that genetic makeup fits well. So the real answer is impossible to say. For example, your DNA sequence and your mother's DNA sequence are different. If you ask me why, because even in the earth, 7.4 billion people are now living. If you sequence their DNA, all are Homo sapiens, but no one identical to another one. Even identical twins, they have different DNA sequence, slight differences. During synthesis of DNA, there are some errors also. I shall tell you later on. So your DNA is not just like your parents' combination of your parents' DNA, but obviously there are some conserved region where your DNA and your parents' DNA matched well, but not 100% identical. Sudeep,
Sir,
do you understand?
yes, sir.
Okay, you got the point. Thank you. Nice question. Any other?
Sir, I have a request.
Yes.
If you kindly send the slides to us as soon as possible, it will be very much helpful for us.
Yes, I shall send you all the slides, and also please go through the YouTube video link, which will give you as much as many times anytime you can browse it and revise it. And obviously I shall give you the PowerPoint slides in PDF format.
Thank you, sir.
Yes, important request, and as usually I do, you will get it. Any more? If no more question, any more?
No, sir.
Please try to ask the question, because your teacher is like a living encyclopedia. You can browse it like Google, and classroom is the safest place to ask the question. And asking question is obviously a process of learning and a unique skill of every individual. No? Then thank you so much.